Thanks for joining us today, and hope everyone is doing well, or at least as well as can be expected. Um, I'm sure by now most of you are aware of the unfortunate news of Sam's passing this weekend. This has been a shock to the entire Stanford community, and he'll surely be missed. Thank you, everyone, for your kind words and support. I'll say that honestly, it has been a struggle to host a seminar series that we've always co-hosted together for the last 4 years. And as you have likely noticed over the last months, I've been hosting this seminar alone with the idea that Sam would be joining me once again someday soon. Moving forward, it is difficult to process that this day is no longer around the corner. However, over the last years, I have learned so much from Sam. In 2016, I recall Sam providing a couple pages of his vision of what would eventually become the PHIND Center. And he gave me an empty office and said, okay, now go build it. Over the next years, we built up the center from just him and I to having an entire community with hundreds of investigators within Stanford and beyond. It has been an absolute honor to get the time with him that I've had and learn from one of the very best by anyone's standards. While it will never be the same without him, we must continue his vision together as a community. As Sam has taught us, taking on the most challenging scientific questions occurs over long time horizons and with extraordinary commitment. I think everyone here is committed to the success of PHIND and human health. And this seminar is an initial step towards moving forward, even without him, and continuing the legacy that Sam included all of us as part of. We will continue on today and take yet another step as a community towards achieving the future of precision health Sam envisioned. And with that said, I'd like to ask just to take a moment of silence in his memory and along his vision. So thank you everyone for being here, and thanks for your continued support of the PHIND. As we continue moving forward, we have on a bright note, we have a very exciting speaker that I will introduce today. We have Vishnu Shankar, recently graduated with his master's degree in computer science with a specialization in artificial intelligence from Stanford University. He completed his bachelor's degree with honors in mathematical and computational sciences in 2018, also at Stanford, with his senior thesis on Bayesian networks for incorporating effect modifiers and meta-analysis. In addition, his background spans biology, mathematics, chemistry, statistics, operations research, physics, and computing. Vishnu has published 6 papers and 3 articles in fields including protein structural prediction, comparison of clinical guidelines, cost effectiveness in type 2 diabetes, development of programs to combat mental illness, cancer diagnosis with analytical chemistry, and machine learning and related areas. So we're very lucky to have him speak today, and looking forward to hearing your presentation. So please go ahead and take it away, Vishnu.
Thanks for the introduction, Ryan. I'm really honored for the opportunity to speak here today, and want to thank all of you for joining. The title of my talk is "What is in your sweat and what can it mean for health and disease?" This work would not have been possible without the support and mentorship of many individuals, 3 in particular who have supported me throughout this effort include Dick Zare from chemistry, Rob Tipshrani from statistics, and Mike Snyder from genetics, who have all, who gave me this opportunity to speak before you today. I'd also like to thank our clinical collaborators from the Veterans Affairs Hospital, as well as the other students in the Snyder Lab and in the Zare Lab, who have all been mentors and teachers for me. This work was funded by the PHIND Center, the Precision Health Integrated Diagnostics Center. And so I think it's worthwhile to take a moment for us to understand, or for us to look at what was the vision of the PHIND Center in the spirit of Dr. Gambhir. I've taken some of his words from an article in 2018. It states, the ballooning cost of treating chronic and late-stage diseases has led to a renewed focus on disease prevention or pre-patient care. Preventive healthcare seeks to augment reactive medicine by preempting disease through preventive measures and early detection. Although not a new concept, modern-day technological advances have created a unique opportunity to finesse preventive medicine with personalized data. It's this last sentence which I think is really important and resonates with this entire project. And I believe the sweat analysis that I'm about to show you today is an example or an instance of this unique opportunity and the possibilities. So to give you some context, everyone here has some kind of conception of what sweat is, that either after exercise or when your body gets too hot, and we know it to be a substance that's on the surface of our skin that is watery and a little salty, but sweat actually consists of a lot more. It consists of amino acids, proteins, fatty acids, and various metals. And to give you a sense of numbers, the entire human body, or humans generally have 2 to 5 million total sweat glands. And here on the diagram on the left, I've tried to show the distribution of these glands, where one can see that at the forehead, there's roughly 360 glands per centimeter squared, whereas on the palms, there's 300 per the dimensions, and at the soles of the feet, there's significantly more, where there's 620 per centimeter squared. Each of these glands is roughly 3 to 5 millimeters in diameter. And at any given time, where roughly 5% of these glands are active. And there's two types of sweat glands. The two types are, one, the eccrine sweat glands, and the two is the apocrine sweat glands. And what I'm trying to show in this picture here is that these glands differ by where they're distributed or localized in the human body. The eccrine sweat glands, while they tend to be universally distributed throughout the human body, they're concentrated in the forehead, in the palms, and in the sole of the feet, as I've tried to show. And the apocrine sweat glands are concentrated in the areas where there tend to be hair follicles, and where in your armpits and genitals. And what makes the apocrine sweat glands unique is that these develop during the time of puberty, given where these glands are specifically found, some of you may already surmise that there's a difference in these glands between males and females. So some criteria or some categories in order to compare the eccrine and apocrine sweat glands, I've tried to summarize that in the table above. The first is the size. As this diagram tries to show, the eccrine sweat glands are about 10 times smaller compared to these apocrine sweat glands, where the eccrine sweat glands are 60 to 80 micrometers. And as I mentioned before, the distribution is different. Another difference that's important is the consistency, where the eccrine sweat glands tend to produce sweat that's clear, watery, and salty, whereas the apocrine sweat glands tend to produce one that's oily and mixed with fatty acids and triglycerides. And one can also note that the function is distinct, where the function for the eccrine sweat glands typically corresponds to regulating body temperature, whereas in the apocrine sweat glands, it corresponds to the stress response. So one question or one point is that if you go back in history and try to look for the first instance or the first mention of sweat, we can go back to 350 BC, where Aristotle has actually mentioned sweat, or this is a translated version, but he has referenced the idea of sweat in this manuscript called Parts of Animals. To quote, the translated version, but although the blood cannot get through, the residue of the fluid moisture, which we call sweat, can do so. And this happens when the body is thoroughly heated. Since 350 BC, we have also come to know about the clinical potential of sweat, and it's been mentioned in articles throughout the 20th century. So the question is the following. If we have known about the function and the clinical potential since the time of Aristotle, why is sweat not more widely used to infer human health and disease? And this was one of the core key questions that I grappled with as a master's student and in the course of doing this project. My experiences showed that there's three key barriers that have limited our ability to realize sweat-based diagnostics. The three barriers are: one, biological; two, measurement; and three is clinical. And now I'll describe each of the three. On the biological front, on the left-hand side, I'm showing a plot which is comparing the plasma levels, or the levels of particular ions that are found in your blood. And I'm comparing that to the ions, to the concentration of the same ion that's found in the sweat, where every data point here corresponds to an individual patient. These plots are based on a published study in 2017, which attempted to track, after 20 minutes of exercise, how does the concentration of these particular ions relate between the plasma and the sweat levels? And as you can tell, the correlations tend to be relatively poor. So this is a question that's long-standing, which is, how correlated are the sweat and plasma markers? When do they correlate and when do they not correlate? And given the variability here in the sweat ion concentration levels, it raises the logical question: what is a characteristic sweat profile, and how much do these profiles actually vary? On the measurement front, in just the preparation for this talk, I found that there were over 30 devices that have been published since the year 2000 that all attempt to do continuous monitoring of sweat in order to track metabolites. Although I'm not aware of all of the details on how the devices work, to summarize two of the key limitations. One is that a lot of these devices, in order to generate a stable and reliable signal, require quite a bit of volume for measurement. They require on the order of a couple hundred microliters. And a lot of these devices, or the majority of them, can only track 1 to 2 metabolites. For instance, some of them in the context of diabetes can only reliably track the blood glucose levels. And this is a premise that I hope to challenge by the end of the talk, that there's value to be learned in not just 1 or 2 metabolites, but in trying to monitor a combination of the metabolites. On the clinical front, the only use case that's currently, the main use case for sweat in the clinic is in the diagnosis of cystic fibrosis, where for an infant, in order to test if the infant is cystic fibrosis positive, generally an electrode or a patch is used on the arm or on the elbow region for the infant. And this stimulates the sweat from this particular infant for up to 3 hours. And afterwards, the chloride concentration is measured based on what's accumulated in the patch, and dependent on what the concentration looks like, the infant is either determined to be chronic cystic fibrosis positive or negative. But clearly, this is not a really convenient test. If a test is going to take 3 hours to complete and it doesn't have full reliability either, there's obviously room for growth here in trying to do something that's more patient-centric. So here's a roadmap of what I'll discuss in the talk. I'll first discuss the applications of the sweat analysis to characterizing renal function in a case study of chronic kidney disease patients. In this case study, I'll also discuss how we have traversed some of these barriers that I've mentioned. This will include the problems and challenges, the measurement approach we have taken, and also some of the statistical machine learning techniques we have used to hone in and focus on the important signals in sweat. Then I'll talk a little bit about the biochemistry of sweat, And finally, I will conclude by discussing what we have learned and some of the possibilities in precision health and medicine. So on the case study of applying sweat analysis for characterizing renal function in chronic disease patients, this work was done largely in collaboration with the Veterans Affairs Hospital, with Dr. Sirich, Natalie Plumer, as well as Dr. Tim Meyer, who have all helped in the sample collection, as well as in guiding me in trying to understand the clinical challenges in trying to try these techniques in the clinical setting. And on the genetics front, I'd like to acknowledge the Snyder Lab, as well as the colleagues in the Snyder Lab, for providing the support in understanding disease and biomarker monitoring. So to give some context on chronic kidney disease is characterized by the loss of function in your kidney or damage to the kidney that prevents it from adequately filtering out toxic solutes or toxic metabolites or compounds that build up in your blood. As the figure at the top shows, there's 5 stages to chronic kidney disease, and the 5 stages are largely determined by what percentage of the function of your kidney is intact. And this function is determined by how well is your kidney able to filter out particular solutes from the blood that are known to accumulate and cause harm. These are called uremic solutes. What is notable here is that 2 things. One is that at the 5th stage of chronic kidney disease, which is called end-stage renal disease, is a stage where when your kidneys fail, you need treatment in order to live. And to also, the other point I want to highlight is just the sheer scale of this problem, which is that in the United States, this condition affects 37 million adults, which comprises 13.6% of the adult population. In just this 5th stage, which I highlighted, there's roughly over 726,000 patients that are being treated for end-stage renal disease. The treatment for end-stage renal disease is not very patient-friendly either. In the left, I'm showing a patient who's hooked to a dialysis machine. And dialysis is the process that's used to substitute for those who have limited kidney function. And it's the process of removing the excess toxins and what I call these uremic solutes, or solutes that accumulate in the blood of those without kidney function. And this is clearly an inconvenient procedure. It requires patients need dialysis 2 to 3 times a week, and each treatment session takes on the order of 4 hours. And it's quite expensive, where annually it's estimated to cost on average of $90,000. So the question then is, why do we need a non-invasive approach to assess renal function? And here are 2 critical reasons. The chronic kidney disease conditions are largely asymptomatic until the late stages. And notably, while 37 million Americans, as I mentioned before, have chronic kidney disease, 48% of the individual in these stages 1 through 4, where they still have some kidney function, so these are patients not on dialysis, are largely unaware of their condition. So the question is, can we use sweat, or can we demonstrate as a proof of principle that we can use sweat to non-invasively and rapidly track renal function and identify risk for end-stage renal disease? Now, this is a pretty ambitious question, and I'll only be able to answer a part of it, but I think the part that I answer shows the possibility of our approach. So in order for us to answer this question in a clinically relevant way, we need human samples. So we collected 2 sets of samples. One is what are called healthy samples. And what makes these healthy samples unique is the individuals in this cohort are part of the integrative personal omics profiling study that was from the Snyder Lab. And this is a study that's been going on the order of slightly over a decade. In this particular cohort, individuals have been longitudinally tracked for not only their clinical markers, but for other types of their metabolites, their genome. So it really gives us a chance to relate when we're extending this analysis if what we're measuring in sweat is related to any of these other clinical markers that we have measured. And for these healthy samples, it was something which I had a chance to actually collect a lot of the samples myself. I would rush between my classes as a master's student when I got a note that there was a patient ready in the clinic in order to collect the samples myself. So based on this endeavor, I got to see what are some of the difficulties in trying to bring this technology in the clinical side. And we were able to collect 38 samples from 6-month regular checkups. And the age and the gender distribution is slightly favoring males, where 60% of the patients that were have collected data from are males, and the mean age is around 55. The reason why I've highlighted this is that it's in contrast to the patients whose samples we collected with end-stage renal disease. 27 samples were collected from these patients. The key feature that I want to highlight here is that for all of these patients, the samples were collected before a dialysis session. The reason for this is if we wanted to test in the first place if sweat could be used to track metabolic changes associated with renal function, the pre-dialysis stage is when these toxic solutes are accumulated to the largest degree. So this would be as a first pass to determine if we could even detect any of these accumulated solutes. And in contrast to the healthy cohort, nearly all patients here are elderly males over the age of 70, 75. So the question is, can we non-invasively detect metabolic changes in sweat related to end-stage renal disease? And for this approach, or for any development to become clinically viable, it needs to match 3 criteria at least that I've laid out, or 3 requirements. One is it needs to be accurate enough to identify patients with impaired renal function. It needs to be robust to the intrinsic variability in the sweat profile, which I'll discuss more in a bit. And it needs to be rapid enough to enable sample collection and analysis in a way that can help guide clinical decision-making. So our methodology is a 4-step approach. We first swab individuals' forehead to collect sweat samples. Then we use a technique called mass spectrometry in order to measure the metabolites. Subsequently, we use machine learning approaches in order to identify and focus on the most critical differences between the samples. And finally, we attempt to interpret the changes we have found and link the model with the biochemistry of sweat. So to describe this in more detail, after the recruitment of patients, which I've shown on the leftmost part of this figure, we initially collect the sweat sample for these patients from the forehead and nose. To demonstrate how easy our sample collection process is, I have a glass slide here, and I'm going to show what the process involves. It simply involves taking the slide and swabbing it across the forehead or the nose, and that's it. And the imprint that's formed on the glass slide is what we're doing the measurement on. So based on this imprint, we use a technique called mass spectrometry in order to detect the metabolites from these sweat samples. Now, to not go in too much into the details, but to just give you an essence as to what this technique does, this technique allows us to identify, or it detects specifically, the charged fragments or the charged ionized chemical fragments that are found in the sweat sample. It's applicable to other biofluids too, but in this case, we're using it for a sweat sample. And based on the charged fragments that we're able to detect, it gives us a chemical identifier and an opportunity to identify or to interpret what's actually found in the sample. In addition, other features that are relevant in our experimental procedure here is that we attempt to add an internal standard and do other quality control approaches in order to ensure that we can control for the batch to batch or the variation that's related to more the experimental setup rather than the actual samples or the underlying biology. Subsequently, after collecting these samples, we analyze the chemical fragments using these statistical learning approaches in order to find the critical metabolites that change in relation to disease before we are able to interpret the significant peaks. So as I mentioned, the slide, which is shown on the bottom right corner, the imprint that's formed on the slide is then measured in a mass spectrometry technique called desorption electrospray ionization mass spectrometry. There's quite a bit going on in this slide, but I only want to focus on a couple key items. The first is that very little sample preparation is in fact needed. So after I set up the experimental apparatus and the stage for the mass spectrometry, the measurement for a sample can be done in under a minute. So that speaks to how quickly we're able to go from sample collection to actually being able to study some of the chemical metabolites. The other point that's relevant here is that this technique is a charge to explain a little bit as to what this technique involves. It's a charged microdroplet technique that hits the sample of our surface. And based on the secondary droplets that bounce off the surface of the sample and enter the inlet of the mass spectrometer, that gives us a very rich chemical profile, which allows us to understand what we're actually seeing in the sweat. To give you a sense as to what we're able to detect, we were able to collect samples from 69 subjects in total, where 42 are healthy and 27 have end-stage renal disease. And if we just count or aggregate the number of unique chemical fragments, we are able to detect over 24,600 unique chemical fragments. These are not necessarily, one caveat is these are not necessarily unique metabolites, but they're unique molecular chemical fragments. And it implies that we're detecting on the order of thousands and thousands of unique metabolites. Here, what I'm showing in this plot, and I want to take a minute to explain this since I have a couple others that resemble this, is on the x-axis, we're looking at the mass to charge ratio. So this is the identifier for each of the 24,000 or so chemical fragments that are found in the 69 samples. And here what you can see is I've overlaid on the bottom the healthy spectra, which is found in blue, the average healthy spectra. And in red, I have the renal disease spectra. And when I mean spectra, I mean the distribution of metabolites or chemical fragments. The other thing to take away is peaks that are found to be larger on the y-axis or to have larger mean relative intensity. It means that those metabolites or species are found more abundantly in the sample. So already we can glean from looking at this plot that there's places where the samples don't overlap. So for instance, here you can see that there's a region in blue, here suggesting differences between the healthy and the renal disease. But overwhelmingly, we're able to see that the red peaks are much larger in a lot of these samples compared to the blue peaks, which implies that we're seeing some changes in the chemical fragments that we're detecting. To give you a sense as to how variable what we're detecting in the sweat profile is, if we were to look at all patients, we find that in all patients, we're able to detect 24,600 or so different chemical fragments. But if we look at just, for instance, 50% of the patients, we see that 50% of the patients share less than 30% of the metabolites. They share approximately 6,640 metabolites, 641 molecular species. Why this is an important point is that it shows that the majority, given how steep this curve declines, it shows that the majority of the chemicals that we're detecting in sweat are ones which are found in few of the individuals. And this underscores the variability. And I'll try to investigate or discuss or speak to this point further later on in my talk. So our first question before we try to interpret what are the important differences is, can we detect these uremic solutes or these solutes that are accumulating in the sweat? And indeed, we can. And what you may appreciate from this plot is that the fold change here, I've tried to look at the chemical fragments. So this is the same mass to charge ratio. And I've looked at the chemical fragments in terms of their fold change when comparing between the two sample sets. A positive fold change in this plot references that the metabolite is found in higher intensity in the dialysis patient samples or in the end-stage renal disease samples. And if it's below 0, it implies that it's found higher intensity in the healthy patient samples. So as you can see from this plot, over 94% of these detected chemicals in sweat are elevated in end-stage renal disease patients. And these are patients before dialysis. So it shows a lot of the uremic solutes that can be found in plasma. There is some kind of correlation or reflection of that we're able to find in the sweat. To give you one specific example, I'm going to highlight the example of uric acid, for which I've shown the chemical structure. And uric acid is a low molecular weight uremic solute. And what makes this distinct and interesting is that it's thought to both cause and negatively correlate with the decreased renal function. So as I'm showing here, and I've split the data into a cohort which we have initially used to do the initial analysis and searching for signals, as well as our model development, and into a cohort that we are using to independently validate and vet the signals that we have found originally in the data. Excuse me. So you can find here that this correlation or this elevation in the sweat of end-stage renal disease patients is preserved when you're comparing the training and the validation set. And you can see that there's quite a significant elevation in these metabolite levels. So this really speaks to the fact that the changes that we're seeing in plasma related to these renal disease patients are ones that we can pick up in sweat. However, 24,000 is a lot. We can't look at all 24,000 meaningfully. So the point is, can we focus on some of the 24,000, the ones that are perhaps the most predictive or the ones that are most critical for helping us delineate between the healthy and these end-stage renal disease patients? So in our efforts to build a model, to summarize, the input to this model looks at these relative abundances of these chemicals in sweat across 52 patients. And the output is looking at, we want to be able to predict if the given patient is healthy or likely has end-stage renal disease based on what we discover in the sweat profile. And in order for this to see the light of day in the clinic, there's likely 4 features that I've outlined that I think are critical for our modeling approach to have. One is it needs to have, of course, high accuracy. 2 is it needs to generalize to diverse populations. And hopefully, if we can build a simple and if we can build a model in the right way, we can have a model that teaches us something about what we're observing in the sweat so that we can interpret. And we can also learn a little bit more about the underlying biochemistry. So the technique I've used is one developed by Rob Tipshrani, and he's been one of the guiding forces for me to adapt and modify these kind of techniques. And so this technique is known as the lasso. And what makes this technique particularly appropriate for this problem is that the top part of the lasso, so there's 2 components to the lasso method. The top part is similar to your boilerplate least squares type of method or linear regression methods, which is trying to fit the data or to maximize the accuracy of the fit. But what makes the lasso unique is that it also controls the variance of the model by restricting the size of the model coefficients. And in doing so, we're able to minimize the risk of overfitting. In addition, one of the features that makes the way the lasso controls variance noteworthy is, as I've tried to show in this plot, the coefficients that are selected, and in our case, that references the chemicals or the molecular species that are selected that are predictive of this difference between healthy and end-stage renal disease, is a sparse subset of the total. So that's what is shown by looking at the corner of this plot, because this is one which optimizes the constraint and the features of this modeling approach. So in order to evaluate the model approach, we use a 2-pronged approach. The first is we use an approach called cross-validation, where we iteratively split our initial set of 52 samples into 10 folds, and we try to build the model in 9 and evaluate the accuracy on the 10th fold. This is done both to optimize the model parameters, as well as it gives us a sense in just the model development stage. It allows us to iterate different models and compare to give us a sense as to how well the models perform on the training set. And then we validated the model in an independent cohort of 17 patients. And what you'll appreciate from this table, I'm not expecting you to read all the numbers, but what you'll appreciate is that the accuracy here is on the order of 88% for this training set. And for the independent validation cohort, the accuracy is around 82%. So the fact that these two are reasonably close suggests that our model is can generalize to new populations. One of the points, so from these 24,000 different chemicals, we find that the ones that are most predictive as selected include these 12 metabolites. And what makes this even more interesting is that the 12 metabolites also show a correlation with the length of dialysis. Here's what I mean by that. So here in this plot on the x-axis, I've shown a patient by their status in terms of dialysis. So on the left-hand side, you have a healthy patient. And on the right-hand side, you have patients based on how long has the patient been on dialysis. So this corresponds to 1 to 24 months. This corresponds to 24 to 40 months. And this corresponds to 40 to 89 months. So you can see that the number of months the patient has been on dialysis increases along the x-axis. And the y-axis reflects with the combination of the 12 selected metabolites that was discovered by this approach, with what probability can we predict that the patient is likely to have end-stage renal disease? And what's noteworthy is you'll find that in the early stage, or in the red box, you can see that there's quite a few patients which appear to look similar to the healthy stages. So this shows that the molecular differences that we're observing in the sweat intensify as a consequence of someone being on dialysis longer. So it shows that there is some of this variance in terms of dialysis length that's captured in our model. The second point that I think is really important to take away is this observation was only true when I looked at the combination of metabolites. When I looked at any of these metabolites independently, or some of the other metabolites we observe in sweat like uric acid independently, I found that it did not correlate with the length of the dialysis. So this seems to challenge the point that there is, rather than collecting one or two for which the variability is quite large, collecting or studying the combination of metabolites may be the key for us to figure out what are the combination of metabolic features that change in individuals or that are correlated with an individual's renal function. Now we're hoping to interpret the metabolites in sweat further. And as you remember, there's not a lot of sample on this slide. So it's really difficult with the limited sample volume for us to try to identify the metabolites. So previously I showed how many we could detect, but now is the challenge of identifying and interpreting. So what I attempted to do was to wash the metabolites into a solution from multiple sweat samples and to concentrate up these metabolites and chemicals, and then to see if we could try to observe based on these extracted metabolites, could we try to validate based on the fragmentation patterns that we observe using the same mass spectrometry procedure, can we try to identify or assign the identity to these metabolites? And to give you a sense as to how many we've been able to identify, in total, if you look at the literature, there's around 80 metabolites that have been identified in sweat. And there's over 24,000 that we have detected in sweat. And out of this 80, 29 of those come from this study that I'm actually presenting to you. So it shows that although we have made substantial progress compared to the rest of the field and the literature, there's a lot more opportunity for growth here in that there's a lot more chemicals that we can identify, interpret, and try to understand the significance of. To give you a standard of comparison, if you look in terms of how many metabolites has been identified in blood, urine, and tissue, the reason why I've selected the number of identified metabolites as opposed to the number of detected is that the techniques that are used for detection are actually different. So this provides a fairer grounds of comparison. And you can see that around 5,000 metabolites are detected in blood, while around 4,500 are detected in urine and 200 to 300. So it shows that although we're making progress, there's a lot more. There's a long way to go here. One specific metabolite or class of metabolites that we identified are these 17 fatty acids, or 17 out of those 29 that I mentioned are fatty acids. And in particular, I've highlighted two examples on the left-hand side, which follow the same general trend. So we see in sweat that there's a general suppression of these saturated fatty acids. And I've indicated the structure above of these fatty acids. These are saturated, as you can see, in that the long tail of these fatty acid molecules, there's no double bonds. So it's a saturated fatty acid. And you can see that there is a suppression of these particular metabolites that are found for both the caprylic acid as well as for the lauric acid. But we don't actually detect this change for unsaturated fatty acids. Given the arrangement of our study, it's difficult to infer what this actually means. But if we, before I get to that, one point that I want to mention that also makes this noteworthy is that if we compare patients from the pre-dialysis to the post-dialysis stage, because all of the patients for whom I've shown the data before all come from the pre-dialysis stage. Here I have zoomed in to a particular portion of the chemical distribution or the chemical profile that we get from mass spectrometry. And I've zoomed into this 190 to 200 mass to charge region. And we can see in this zoomed in region in particular, we can see that the intensity of these metabolites don't change significantly, suggesting that these changes are quite robust. So if we interpret this, several published studies report changes in fatty acid breakdown related to chronic kidney disease in blood and urine-based analysis. But what is also exciting is that other analysis that's been done in blood shows that fatty acids are not really dialyzed by this procedure. So the sweat analysis corresponds in this sense with the dialysis, where it finds that these fatty acid profiles or differences are largely unchanged when you compare before and after dialysis. There's still two questions that we need to answer or address, which is, one is, how much does a sweat profile vary? I've alluded to this point a couple times. And does our approach actually incorporate this variability? So to understand how much does a sweat profile vary, we collected from the same 6 patients' replicate samples, but 8 months apart. So we collected samples in November 2017 and June 2018, and these are all from end-stage renal disease patients. And this plot has the same format as before, where you can see the mass to charge ratio on the x-axis, which provides a chemical identifier. And on the y-axis, you can see the relative abundances of each of these chemical fragments in the data. And here we find that although there's overlap in some areas, there's differences. For instance, you can see areas where just the black peaks, which is the ones from November 2017, stand out. In other areas, the yellow peaks stand out. So there are differences here. The question is, can we quantify what is the nature or how different are these two profiles? So one way to quantify, this isn't the only unique way, but this is one way to do it, is if we look at which peaks are low variance and high variance, and I categorize this or stratify these metabolites as follows. A metabolite is considered low variance if the average fold change is less than 2 between these replicate samples. And we find from this analysis, 60.9% of the peaks have high variance, which indicates significant variability in the sweat profile. If we delve deeper, we can find that the area in which these low variance, these 39% low variance peaks are concentrated, happen to be in this 200 to 300 M to Z region, and this happens to coincide with a lot of the fatty acids I showed you before. So it suggests it doesn't change, that it's relatively invariant between samples, and it also doesn't change after dialysis, which highlights how robust it is. Next, we can ask is, do these metabolite levels correlate with the plasma levels? And I have tried to summarize a couple studies. The top 2 rows correspond to our studies, and the bottom 4 rows correspond to other peer-reviewed published literature. And you can see in this column, which looks at the correlation coefficient, that our study shows that we don't correlate very well when we look at the values, these uremic solutes in the plasma, and when we compare the correlation of, when we compare those particular metabolites with what we're observing in sweat that's associated with renal disease, we find that the correlation is not very good. We do find that there's other metabolites that have been measured in these other studies, but what I want to highlight here that I think is an important point is this is the only study that I could find, or one of the only, that characterizes the sweat profile without stimulation or continuous device monitoring. This is distinct from all of these other studies I've shown in this table, and most of the other studies you'll find in the literature, where the sweat to plasma correlation appears poor. For instance, in this 5th row of the table, if you compare plasma and potassium, you can see that the correlation is abysmal here too. So it appears that stimulation plays a role. The stimulation of the sweat glands plays a role in the correlation between plasma and sweat. So this raises the point, what does the resting sweat variability look like? To further investigate this question, here's what I tried. I looked at each of these 6 patients, these replicate samples that we collected from the same patients, and I calculated the percent difference in these samples in those 8 months. So I calculated based on the measurements that we had, what is the percent difference in these measurements? And we find that the percent difference, there's a couple things which I want to point out here. The first is that in some cases, the combination of these sweat metabolites has a smaller variability compared to the plasma urea. For instance, you can see this to be the case in patient 2 as well as patient 4. And the other point which I want to mention is that the combination of these metabolites has similar variability to plasma urea, where the average is around 23.6% for plasma, sorry, for the sweat metabolites. And for plasma urea, it's around 15.6%. The other thing is this trend does not hold when we look at the individual metabolites. So this further seems to strengthen the point that there's value in our approach in looking at the combination rather than the individual approach. So even though the combination, as I show in row 2 on the last slide, doesn't correlate very well with plasma, it is the case that it's at least internally seems to have some kind of self-consistency. So to summarize, we were able to cross 3 barriers for sweat diagnostics in this project. On the biological front, we were able to figure out that we were able to elucidate that 39.1% of the detected chemical species in sweat tend to be low variance, suggesting that there's significant variability, and that the significant fatty acid perturbations and these uremic solutes can actually be identified from sweat. And we found that these fatty acid changes were robust in that it was preserved before and after dialysis, as well as the fact that these peaks appear low variance when we track the same patients. On the measurement front, the 2 points which I think are important is, one is that the combination has its strengths, which 1 or 2 metabolites cannot capture. And the second point is that we're able to detect over 24,000 molecular species based on small sample volume, which presents an enormous opportunity. On the clinical front, we were able to make 4 advancements. The first is that we have a rapid and pain-free approach. As you saw, it took less than 30 seconds, and I wasn't in any pain, and none of the patients were, in order to collect these samples. And it's also accurate. We find that it's reasonable. We are able to achieve around 82% accuracy. And that the signatures that we pick up are able to correlate with the length of the dialysis treatment, and the variability of what the signatures we detect is similar to plasma. That's not to say that this is a complete or a perfect effort. There's still outstanding challenges. And I hope this talk will motivate efforts to study some of them. On the biological front, our data is suggestive that the stimulated sweat differs from the resting state, but it's not clear how this differs. It's clear that age, gender, exercise, diet, and change in climate all affect the sweat chemical composition and the rate at which we sweat. But what does this mean in terms of not only the composition, but in terms of disease? It's difficult to do a controlled study, but this analysis is vital for us to get a sense of the baseline variability. On the measurement front, how can we reliably monitor a combination? And on the clinical front, can we figure out what affects this plasma sweat correlation, and can we better complement current clinical markers found in blood? I started this talk talking about the precision health and medicine paradigm, and I want to revisit this paradigm in the spirit of Dr. Gambhir, particularly the part in blue. And I hope you take away from the fact that this is really a unique opportunity to finesse preventive medicine with personalized data. And specifically, as I've shown on the right-hand side of this plot, human metabolism is extraordinarily complex in this network of interconnected pathways. And for us to be able to tap into this or learn about this non-invasively, I think our efforts speak to the core themes of PHIND, where we're able to show the integration of analytical chemistry, human metabolism, imaging, and machine learning are powerful for advancing this diagnosis. And none of these efforts were very much facilitated by PHIND and very much facilitated by PHIND and in the same theme as what PHIND has to offer. So I can take any questions that the audience may have.
Great, thanks for an excellent talk. So as usual, feel free to either raise your hand on Zoom and we'll unmute you, or you're welcome to also write your questions in the Q&A. So I'll kick us off with one that was already placed. Given that you touched on this a bit on your challenges slide, given the age difference between the controls and renal patients, can these results really be ascribed to renal failure, or can they just be reflecting a non-specific aspect of aging?
That's a very important question, and I'll speak to this from two levels. The first is that in an independent cohort, and this is a study that was done separately from my own work, in an independent cohort on 194 sweat samples that were studied, certain metabolites were found, particularly phospholipids, were found to correlate with older patients compared to younger patients. And so one clue that suggests that it's not just an effect of age, ethnicity, and gender is the fact that our selected combination of sweat markers are independent from what has been found to relate to gender, ethnicity, and age. So that's one clue. The second, which I think is a good point, is that we are able to find that the uremic solutes that we're specifically detecting, that have been detected in plasma, are also found in blood. So this is another clue that if it were, that maybe that there's an age-related cohort, but age-related factor, but we're actually able to detect features that are really associated or are actually found in the plasma, and we're also able to detect them in sweat. So that provides two different clues or responses to say that what we're detecting is not just attributable to age, although it may be one of the components that contributes to the differences.
Excellent. Looks like Robert, you have a question. Please proceed.
Yeah, very nice talk, Vishnu. A couple of things. There was, as you showed, there was a hint that the markers were correlated with how long dialysis had been going on, but it wasn't that inclusive to me. Why not just try to predict in a direct way, build another model that tries to predict how long they've been on dialysis? And the second question, oh, sorry, but let me just,
okay.
The second one's kind of related, is that it seems like the clinical, I didn't quite see what the clinical question was, but it seems like an important clinical question would be to try to figure out someone in an early stage, whether if they try to detect that they have a high probability of going into end stage. So why not, can you collect a time course of these samples from patients in different stages and use the changes in those metabolites to predict the probability of going to end stage? That would seem to be a direct.
Yeah. Related to the first point, I agree. That is something that can be directly measured. I just wanted, as a first pass, before we try to fit a model to dialysis length, I wanted to initially see if there was a difference that could be detected in terms of just looking at the groups of patients as a whole. And so this is something that I can do and I should do, so that's a good point. And related to the other question, as I mentioned before, you are right that the time course is what is needed in order for us to answer this question. But in order to do that, I need to make the point that no one has actually looked at sweat in the context of renal failure and characterized its variability in the way we have done before. So even before we could start to look at time course data, which is more complicated, it would be good if we could at least, we wanted to first be able to see if we could even detect it at the end stage renal disease level. So that's an excellent follow-up that needs to be done. But this I felt was a prerequisite in order for us to be able to get to the question, which is a more ambitious and complicated one, as I tried to allude to earlier in the talk.
That's a good point. If you had not been able to discriminate healthy from end stage renal, then that would have been
Correct.
made the second part of this thing impossible. So it's a good first step that was necessary.
Correct.
All right, great. Next question is, are some of the metabolites you detect known to be produced by bacteria that live in skin? And if so, since they differ in bacterial species, are they likely to occupy distinct geographical regions of the skin? And to what extent does the metabolic composition of sweat differ in different regions of the skin?
That's a very good question. And it's one which is still not known. So for instance, I made this distinction in the beginning between the eccrine sweat glands, which produce watery type sweat, versus these apocrine sweat glands. And in addition, some of the areas where the apocrine sweat glands are located, for instance, in the armpit regions, are also known to have some of these bacterial species that correlate with some of these metabolites. It's something which has not been very well investigated, and is something that's worth looking into. But there is a correlation that has been found between the location of the sweat glands that are found throughout the body versus the bacterial species that are specifically found. Whereas in some areas where apocrine sweat glands are found, we find higher proportion of these bacterial species, not higher proportion, but we find these bacterial species are more predominant compared to other areas. But it's a question which is still largely open.
Sure. Can you talk a little more in detail on the sweat collection? So they say, I assume the volume is different when you scratch each time. Can you describe how you normalize the volume and the final MS signal intensity?
Sure. So let's get to that. So a couple things. So for starters, the first way in which I'll speak about it in the experimental first, then I can talk about sample collection. So on the experimental setup, we first add an internal standard. The internal standard is spiked into the electrospray solvent. That is what generates the drop, what is partially responsible for generating the droplets that impinge on the surface of the sample. And the internal standard allows us to normalize for volume between samples because it gives us a sense for the relative intensity that we're observing in the sweat profile. What is the concentration of that particular internal standard that we have added into the solvent? So we're able to normalize for those differences in some capacity based on this internal standard. The second point is that every time I collect a sweat sample, because I've collected a lot of these samples and done the measurements, there are a few characteristic peaks I'm looking for, or metabolites I'm looking for, to get a sense as to whether the sample has something, has a large aberration, or whether the sample is typical or close to some of the other samples that we have seen. So particularly, I'm looking for these saturated fatty acids. So I look for palmitic acid, which is one of the fatty acids that I know to be one of the fatty acids that dominates the sweat profile and relatively invariant in comparison to the relative intensity of the other metabolites that are found in the profile. So first is I'm using the internal standard to normalize for this batch to batch variation. And the second is there's some kind of quality control based on the fatty acids. And this also gives me a sense if there's some kind of skin contamination or possibly other kind of contaminants that have emerged in the sample collection that are reflected in the spectra. Now, apart from this, apart from just the sample collection I showed during the talk, there's no real, we just cleaned the slides, but the only additional thing that we request is if a patient or a subject has some kind of cosmetics or makeup, we ask them to, we wash out those cosmetics and makeup, and then we collect the sample. That's the only instance where the sample collection is distinct from just what I showed in the talk.
Sure, just a related question. How much sweat can you collect with a glass swab from a patient who is not sweating?
I don't know. I don't know in terms of the volume, but I think our talk shows that that the imprint that we're getting from the slide is enough to detect all of these metabolites. So even if the volume is small, the signal seems to be enough for us to be able to characterize and figure out these metabolic differences.
Sure. And do you plan to compare metabolites in sweat with those in urine, which can be obtained in larger volumes and non-invasively obtained?
Yeah, that's a good point. So a lot of these uremic solutes that I've mentioned several times in the talk are found in, we have shown that it's found in the sweat, but we can also track the clearance of those particular metabolites by looking at the urine. It's something that we hope to collect, and we can actually compare, for instance, uric acid we can actually compare uric acid and some of the other amino acids that we're able to detect from the sweat profile. We can compare those levels to urine. It's not there in the talk, but it's something that can be done and should be done. So I agree.
Excellent. Let's see. Any other questions for our speaker? I guess I have a question. So you mentioned, so it's interesting that, you know, in this case, having additional metabolites definitely was beneficial for you. And it looks like you're still investigating a number of different ones. How many metabolites are do you think is reasonable to measure, assuming that the combined measurement of these different ones, you know, is there like an upper limit to how many you would like to gather data on? Or because usually we think of trying to have like the minimum number of like viable measurements to get an accurate, you know, prediction or whatever it may be. You know, how do you view this, and what kind of, you know, scale of measurements are you looking at?
Well, I think given if the measurement approach and the approach that we have used to build the model, so that's one of the reasons I highlighted the sparsity of the modeling approach that we use, in that it's able to drastically reduce the initial number of metabolites that are detected into just a couple. We found that we were able to select just 12 from over 24,000. And so in terms of how many is a reasonable upper limit, I think that will actually depend on the heterogeneity of the data. So if it happens to be that individuals with a particular phenotype have large variability in their profiles, then obviously more metabolites are needed. But I don't think it makes sense to put an arbitrary upper limit in the combination of metabolites that you select, because you're going to lose important parts of the signal. As I've mentioned earlier in the talk, when you look at the individual signals of these 12 metabolites, you find that it's limiting. But when you look at the combination, you find that the signal appears more robust. So I think this kind of limitation of what is the upper level of metabolites is a little arbitrary, but as long as the approach to doing the sample collection, the experiment, and the model development and deployment are rapid, I don't think it matters that much. I think the accuracy matters more, as well as the utility it has in the clinic. So it'll matter more on the clinical utility. And I think that the approach we're using is well poised to do what you state, where it can focus on what's most critical while capturing a majority of the signal in the data.
Yeah, I guess what I was thinking more in terms of, I understand what you're saying, but in terms of like practical terms. So, you know, is there a certain threshold in terms of deployment into the clinic that would make it cost prohibitive, or those types of aspects? So I'm thinking, you know, you have a solution, I want to deploy it, you know, would you be able to have whatever it may be if that's the best
Yes.
possible
Yeah,
model?
the prohibitive part of the cost is in trying to get a portable mass spectrometer that's in the clinic. That is the prohibitive part of the cost. The other components are not that prohibitive in that are not the limitation. So if there is something that if we can get a portable mass spectrometer that's clinically accessible, then if the entire procedure that I've shown here can be done quite rapidly. So I don't think there's a cost issue associated with any of the other elements. I think it's just of the device or the machine itself that needs to be brought in a setting that is complementary to the existing clinical care.
Sure. Okay. We'll do one last question. So what is the next stage of this project?
So the next stage of the project, I think, is twofold. One is to look into the urine analysis that was mentioned by one of the questions earlier, and to also look to figure out some of the confounders that affect this resting sweat composition, whether that be sweat that is stimulated or sweat that is caused by exercise versus sweat that's baseline, and to first do a comparison for the same metabolites that we detect in our study versus external studies. A little bit more long road is along what Rob Tipsharani said earlier, which is if we can actually use this approach to do something that can directly have an effect in the clinic, where we can rapidly screen chronic disease patients by collecting samples in a time course manner and use the information that we obtain from the sweat sample in order to predict who may be at risk, that is the natural extension of the study, because that's more similar to how such an approach may be useful in a clinical setting.
Okay. All right. Well, thanks so much for a very clear and excellent talk, and you know, very interesting insights on sweat and analysis. So thanks everybody for joining us, and have a great rest of your day. Thank you.